I'm very strict with time, as you know. We will have a very, very good panel discussion later, and you'll have the occasion to meet Paula again. And now that you are awake, because that's important, I think of these events a little bit like sports. We are practicing an innovative sport here. So you have to be awake for it, meaning we expect equal participation in the audience and up here. Now, given that, I'm extremely happy to introduce my eminent colleague, Associate Program Director Marcus Dulloff, for the startup proportion of this event. And one thing I wanted to say outright is that one of the most exciting things with this particular format is that we get some startups that are very early in their formation, and I think we have a couple of stealth candidates here that haven't really been on stage before, and that's always very exciting, particularly in a field like cancer that I think means so much to all of us, both professionally and some of us also personally. So with that, Marcus.
Thank you, Trond. So like Trond said, we're going to build on this emerging trends theme, and we have 8 startups that I'm going to introduce, and these are truly emerging next phase technologies likely to be disruptive, and some are even emerging out of stealth mode today. So you'll be the first to see them. You'll be able to say that I was there when, and again, to Trond's point, you can even say I was there then, and I made a connection, and we made a partnership at this event. They'll each talk for 3 minutes. There'll be no Q&A immediately following. The intention is that you're going to follow up during the networking session afterwards. Our first speaker is Cliff Reed, CEO of Trevorrow. He holds a bachelor's in physics from MIT. And the title of his talk is A New Approach to Personalized Medicine for Cancer. Stay here. Cliff.
Thank you very much, John. Also Harvard MBA and a Stanford PhD, but we don't talk about that. So I am indeed the CEO of Trevorrow. Trevorrow is a brand new company just founded this year, a spin out of the Scott Manalis lab, founded by a bunch of MIT engineers and a bunch of Dana-Farber cancer researchers, and Rob Kimmerling is one of the founders in the room with me here today. So I'm going to talk to you about our new approach for personalized medicine in cancer. So one of the key challenges in personalized medicine for cancer is the lack of predictive biomarkers. We just don't have very many of them right now. In fact, MD Anderson did a study in the last couple of years where they did deep sequencing of 2,600 cancer patients, and they found that fewer than 7% of them matched any therapy associated with their mutations. And when given therapies, about 1.5% of those people actually benefited from the therapies. So we're a long way away from good predictive biomarkers in cancer. Now flip over and look what happens in infectious disease. Infectious disease has these amazingly predictive biomarkers. It's called culture the bacteria, hit them with a bunch of antibiotics and see which ones work. It works for about 98% of all patients with bacterial infection. So the question is, can we do in cancer what has been enormously successful in infectious disease? And the answer is yes, we can. So Scott Manalis and his team have invented a new device. It's called the Suspended Microchannel Resonator. And what this device does is it extremely accurately measures the weight of a single cell and the changing weight of a single cell. And the reason this is important is because they discovered that when you take a cancer cell out of a human body, immediately while it's still viable and you hit it with a drug, it shrinks imperceptibly. If the drug doesn't work on the cell, it doesn't shrink. So for the first time, we can actually measure the biophysical property of the cell, not the biochemical property, to see if it responds to the drug. And what we do is run a cell through a whole series of these measurement devices to measure its changing weight. So this is a biophysical biomarker that potentially can be applied to a large number of cancer patients and a large number of cancer drugs, approaching a universal biomarker. So we've taken this technology out of the clinic and into, don't think that's going to work. That's all right. Out of the clinic and into patient testing, where we did this with Dana-Farber, our partner, and ran this against 9 patients at Dana-Farber and showed that we could accurately predict the results for all 9 of those patients. We're about to fire off a 100-patient study at the Farber. The other interesting thing this technology can do is sort cells by their resistance to drugs, sequence them, and figure out which cells are resistant and which cells are sensitive. So we've only partnered with the Farber right now, and very much interested in partnering with companies like yours to both explore the universality of this biomarker and its use for drug development. Thank you very much.
Thank you, Cliff. Our next speaker is Todd Garin, co-founder of Honeycomb. He's was a postdoc associate at Professor Love's lab at MIT, and his talk is Supercharged Single-Cell Profiling Technology. So that one. Yep.
Oh, we are at the end here. Can we get back to the... Anyway, I'm Todd Garin, the CSO of Honeycomb Biotechnologies. So Honeycomb focuses on providing tools and service for what we call supercharged single-cell profiling of clinical samples. And it's been spun out of technology developed in Dr. Chris Love and Dr. Alex Shalick's lab here at MIT. And they are 2 of the world's experts in single-cell profiling. So what we see as the foundation of the precision medicine that's coming is really going to be the interconversion of biological and digital information. So you start with a biological sample. You need to convert the biological information present in that patient's samples into digital information so they can be effectively queried to really understand nuances of every particular person's disease. And then what you also have are extracted from healthy cells instructions that allow you to re-engineer replacement parts for each individual's disease that can be reintroduced into that patient to fix the particular problem with the biology in that patient. So Honeycomb focuses on the reading aspect of turning biological information into digital information, and we really think single-cell profiling is the way to maximize the amount of data recovered from a given biological sample, primarily because cells are the fundamental unit of biology, so they are what drive biology. However, there are many challenges to applying this in the clinic, particularly being able to do this in any kind of clinic worldwide. How do you do this in solid tissues where you have to potentially dissociate the tissue? Really having the expertise to do these techniques in the clinic is very challenging to have that in every clinic, as well as high cost currently. So our solution is what we call the Honeycomb, the Hive Array, which I have an example, but I double fisting here. The Hive Array is a planar array of up to 1 million micron wells. And we can use this kind of as an ice cube tray for cells, we call, that can capture tens of thousands, 100,000 of cells in these and be able to analyze at the single cell level both single cell protein and RNA content. And we really think this has 2 fundamental advantages over competitors in the space, particularly in the droplets space. One is the simplicity, so there is no complex microfluidics involved, and so there is no capital equipment required to do this technique, and so all you need in the clinic is a single array that can be sent worldwide, and we've sent it to 6 continents already. The other is open addressable wells that allow you to do stable storage. And so to get into stable storage, what we can do is robustly recover mRNA from fixed cells and tissues. And what this allows you to do is simplify the clinical workflow, centralized processing, and be able to do long-term storage for retrospective target discovery. And so we are currently looking for alpha testers to use either our profiling tools or as well as a service that Honeycomb will provide. And we'd love to talk to you after.
Thank you, Todd. Our next speaker is Laura Indolfi, co-founder and CEO of Panther Therapeutics. She was an entrepreneur in residence at the MGH Cancer Center and postdoc associate with Professors Ellman and Langer at MIT.
Okay, thank you. Good morning, everyone. We are one of the spin-out of the Coke and the collaboration between engineer and physician. And the idea came actually from one of the bridge projects where we were put all in a room with the oncologists at MGH and engineer at MIT and said, we need to do something for cancer because specifically in certain type of tumor type, we are really underperforming compared to what the rest of the world is doing. So cancer is usually seen and treated as a systemic disease, and as such is treated with systemic injection. So regardless of where you get your tumor type, you get a bolus injection that will reach everywhere into your body, and as a result, you will have high toxic effect in the perfectly healthy tissue that you still have, and very little therapeutic effect at the tumor site. And for specific type of tumor in the gastrointestinal tract, for example, it's actually the primary mass that creates all of the problem, that allow the patients to be sick, and that most of the time it's also the leading cause for the mortality. So we need to do something to change this. And as an engineer, we have been looking at, can we parachute the drug directly where it is needed? We have talked a lot about personalized medicine in terms of, or precision medicine in terms of personalizing it to the patients, but can we do some precision medicine in terms of delivering the drug precisely where it's needed, so that you can increase the therapeutic effect to the tumor site, decrease the side effect to the rest of the body, and in a full 360 degrees, hopefully also provide the mechanical containment of the primary mass so that there is no spreading into other organs and no metastasizing. We have been working for 2 years almost right now, and that's what we have come up with. The first product of Panther is an implantable patch. It can be folded, and I, again, cannot do multiple things with one hand, but it can be folded to fit perfectly into the laparoscopic tools that there are surgeons are using to staging tumor to see if they are resectable or not. We have engineered it to fit the problem and solve it. So the polymer is biodegradable. Once it's implanted, it will degrade over time and allow for the delivery of the drug only at the tumor sites. It's a unidirectional delivery. It will go only into the tumor sites and not in the backbone. And we have showed in a very complex model that we are able to overcome all of the challenges and limitations of systemic treatment. We increase the amount of drugs that is delivered at the tumor site. We extend drastically overall survival. We have a 100-to-0 rate between the control group of systemic IV and localized treatment, and we delay the time of metastasis of the patients. So what can we do in situation like this of partnership? We have a technology that is superior to the current standard of care, and it's very helpful for pharma company to repurpose drugs that have been shelved because of systemic toxicity. We bypass completely the accumulation in the liver, in the kidney, and in the spleen, so drugs can be repurposed. We can enter into hard-to-reach location, but also for the medical device company, there is a hot buzz to enter into interventional oncologist, and we will be the perfect partner for that. So thank you very much, and hope to chat with you later during the break. Thank you.
Thank you, Laura. All right, so our next startup is one that recently came out of stealth mode, so not exactly stealth. There'll be some others that did or are coming out of stealth today. Ulrik Nilsson is the CEO and co-founder of Torque, co-founded with Professor David Irvin from MIT, and he'll be talking about cancer immunotherapy 2.0, tackling hostile tumors.
Thank you. It's a lot of fun to be here. This is my second MIT spin-out. The first one was one called Merrimack, where I was the chief scientific officer for a number of years, also located here in Cambridge. The next one is Torque. We're pioneering an entirely new approach to cell therapy for cancer. We do that through a methodology we call deep priming, which we think is going to be revolutionizing the treatment of cancer. Unless you've been living under a rock for the last year or so, you will have seen the results of some of the CAR T therapies and their application to ALL and lymphomas. They've shown spectacular results where in some cases curing more than 80% of the patients with a single infusion of a T-cell therapy. That has changed forever the face of cancer and cancer drug development. The big challenge is how do you take this to more patients? And that's where Torque comes in. We're trying to move this into diseases such as solid tumors where responses with cell therapies have been incredibly rare. When you talk to people in the field, no one thinks that taking something like a CAR T therapy to more cancers is just about finding the right target. Really, the challenge is to overcome the immunosuppression that's present in the tumors and in the patients that's limiting the efficacy of these cell therapies as you take them more broadly. At Torque, we're developing a technology called deep priming that allows you to do that. We can really alter the microenvironment by attaching nanoparticles of biotherapeutics to the outside of cells that post-infusion into the patient will release into the microenvironment, and through that overcome some of those immune suppression and broadly engage the immune system in the fight of the cancer. Here on the left-hand side is shown a little movie of a cell carrying one of these backpacks, as we call them, over to the tumor cell. The backpack is blue. You can see how it engages with the tumor cell, and then you sort of see the backpack spread out at the interface with two different cells. This is the ultimate microdistribution of a drug. It really gets to the interface where you want these things. On the right-hand side, you see an actual efficacy study where we're treating tumors with T cells that are reactive against the melanoma, and we put an IL-15 cytokine backpack on them. And you can see how the backpack radically changes the efficacy of this cell therapy. We're using this deep priming approach in a number of ways. One is we're taking our own T cell therapeutics to the clinic in the first half of next year. We're also actively partnering with other cell therapy companies for which we can do deep priming to make their cell therapies succeed where they can't today. I'd be happy to talk to you guys about how to do that in collaboration. Thank you.
Thank you, Ulrich. Next speaker is Jason Norris, co-founder of Electrify. He holds an MBA from the Sloan School of Management and is co-founding the company together with a PhD from MIT from the Department of Aeronautics and Astronautics. And so logically, he's going to be talking about applying rocket science to biotechnology.
That's obvious that I'm going to be talking about applying rocket science to biotechnology. So as Marcus mentioned, I'm Jason. I did my MBA here at MIT. And my co-founder, Chase, was working in the Space Propulsion Lab on the propulsion system for cube satellites in low Earth orbit. And we decided that it made perfect sense to go from that to starting a biotech company. Our team now is 8. We have 5 PhDs or postdocs from MIT, along with 4 professors from MIT on our board of advisors. Daniel back here did his PhD in biology at MIT. He's the only guy wearing a tan jacket here, so feel free to talk to him at any point in time as well. So what exactly are we doing? Within the monoclonal antibody space, delivery is a major problem. If you're a cancer patient, you're going to go sit on an IV for an hour to 12 hours to get these infusions of monoclonal antibodies. It's a big issue for both the patient and for the payer. Payers are paying about 3.5 times what the infusion center or hospitals are actually paying to the biopharma companies for the drugs themselves. So what we do is we take this multi-hour IV infusion to subcutaneous delivery utilizing our proprietary formulation technology called Electrojet. This is good for the patient. It's also good for the payer. You may say, what is this magical process called Electrojet? What we do is we create colloidal suspensions of microparticles utilizing an atomization technique. So in layman's terms, we're creating dense spherical proteinaceous balls that can be injected into the, that's the layman's terms. But we're creating these spherical balls that can be injected into a patient, and because of the sphericity of the balls, along with the decrease of the intermolecular interactions that our formulation technology provides, we're able to get these balls into the patient without the aggregation that usually would occur under normal monoclonal antibody conditions. What this results in are concentrations up to 500 milligrams a milliliter at 20 centipoids. We're actually closer to 10 centipoids. We're able to maintain full bioactivity without denaturing the underlying protein. We're compatible with a host of biologics utilizing GRAS materials, and we're able to scale the process. So our pilot scale system that we expect to have operational in the next couple months is able to process multiple kilograms a day. And to the extent we want to scale that up, we have the ability to do that with multiple machines. As far as partnerships go, we're in collaboration with one biopharma company now. We expect to have a second partnership signed up in the next week and a half. We're happy to talk to you about this. The technology itself belongs to us. We filed for we own all the patents ourselves and work with a company called Clark & Elbing as basically in-house counsel here. So It's my contact information, and again, happy to take any questions later.
Thank you, Jason. Our next company is coming out of stealth today. Patrick Stern is the CEO and founder of Sextant. He holds a PhD in biology from MIT and was a postdoc at the Koch Institute. Harnessing Cell Death to Drive Immunity is the title of his speech.
20 years, most recently, my company was doing sponsored research at the Koch Institute in Forrest White's lab. So immuno-oncology is focused primarily on the immune system. What can we do to the immune system to make it more active? The tumor itself is largely ignored. How can we leverage the tumor to provoke an immune response? Why is the tumor ignored? Well, traditional textbook thinking says that dying cells, specifically apoptotic cells, suppress immune responses. There's a lot of data to support this, but that idea is incomplete, as I'm going to tell you. Technology is also lacking to study cancer cell death, and that's where our platform comes in. This is really a missed opportunity, ignoring the tumor cell death. If we can harness cancer cell death to provoke potent cancer-killing immune responses, we now can accelerate what we're already doing with cancer immunotherapy. So our platform is a next-generation preclinical systems pharmacology platform. It's a bit of a mouthful. It's essentially two technologies. One of these is a gene expression technology. It's the fastest gene expression system in the world. It lets us, in this case, looking at tumor immunology, kill cells precisely through different apoptotic pathways. Doing this, we've been able to identify which types of apoptosis can actually prime cell-killing immunity. We've partnered this with a mass spec approach which identifies newly made proteins. So we can capture and identify the proteins that are made as cells die in tolerizing versus immunizing conditions. So we've cracked the biology of cell death. We've identified the signals and the conditions that either drive immunity or suppression coming from dying cells. Here we're using an immunizing cell death vaccine where we have normal cells that are loaded up with melanoma antigens. Here, the blue line, sort of classic apoptosis, which doesn't really have a whole lot of meaning, but that's what you would expect. Apoptotic cells do not initiate an immune response, and so then these mice die very quickly from melanoma challenge. Here we've killed cells different ways in the red line, also through apoptosis, but with some particular manipulations, and now we get potent, durable, cytotoxic immunity that rejects melanoma, and these mice are tumor-free, except for two, one at 3 months, one at 4 months, that do not develop tumors. So we're looking for development partners to take existing oncology assets, which really have no measure for are they immunizing or not, and turn them into first-in-class, tumor-focused immuno-oncology therapies. Pharma partners would bring existing oncology assets. We would bring targets, assays, diagnostics for immunizing cell death, and a site license for our next-generation discovery platform. I'll be out there, and I would love to speak with some of you about how we can help you develop new therapeutics for immuno-oncology. Thank you.
Thank you, Patrick. Our local labs is the next startup also coming out of stealth. Carlos Castro Gonzalez is the CEO and co-founder of that company. The company was founded by 4 MIT postdocs, Carlos being one of them. And he'll talk about non-invasive white cells monitoring device for chemo patients.
Thank you for the introduction, Marcus. This company was actually founded recently, 2 months ago, and we are spinning out a technology that we've been working at MIT for 3 years. And we are particularly developing the first non-invasive device for white blood cell monitoring. Part of the motivation for this research started 3 years ago when one of my best friends from Madrid was diagnosed with a non-Hodgkin's lymphoma. And one of the things that I remember more vividly about this experience is that he was really worried about the status of his immune system and the possibility of getting an infection. And this actually happened because as a side effect to the therapy, the patient's immune system, and particularly their neutrophils, which is the most common type of white cell, go down to low values that if sustained for too long could put him in high risk of a life-threatening infection. This is a major unmet medical need for these patients, and this year alone in the US, 650,000 patients will start chemotherapy treatments, and approximately 1 of 6 will get one of these infections while being immunosuppressed. This obviously has really negative clinical outcomes for these patients, but also a high cost to the healthcare system at $25,000 per case. We wonder why this need wasn't being met by current technologies, and one of the major limitations is that all existing solutions require a blood sample, and therefore patients must travel to a healthcare facility, and that limits how frequently this testing can be done. That's why we started developing a completely non-invasive technology that would enable more frequent monitoring that is currently possible with any technology, and that would allow to detect these patients when they are critically low in terms of immune system before they develop the infection, giving precious time for clinical intervention. So at MIT we developed a microscopy system that looks through the skin in your finger at this area called the nail fold. That's a very unique part of the body, because capillaries are very superficial. That was meant to be a video, so one of the videos that we collect, so sorry that is not working, but the data that we collect basically are videos of these microcapillaries that are the black loops. It's coming, all right, okay, so you can see it now. So you can see those black loops and the blood flowing through those microcapillaries. They are so narrow that the cells must squeeze through them one by one. And because of the illumination we are using, red cells absorb the light, white cells do not, so we observe them as those gaps that you can see in the images. From there, we develop algorithms that detect those events and get an estimation for the immune system for these patients. We actually tested this at the Mass General Hospital and in chemotherapy patients before and after they receive chemo, and showed that we can detect those with critically low values with 95% accuracy. With that milestone achieved, we are excited to be working on our product development in terms of hardware and software for a product that will give results in 1 minute. And from our market research, we have identified different value propositions and customer segments. We are focusing as early adopters for hospitals, and we have a couple signed letters of intent. And we estimate that we can reduce 50% of their chemo-related hospitalizations. We are also focusing on pharma companies as a way to use the drugs more safely and effectively. So we are looking for partners willing to engage in co-development of early technologies for validation pilot studies. So please come talk to me after this event. Thank you very much.
Thank you, Carlos. So last startup today, Alec Nielsen, co-founder of Asimov. He holds a PhD from the Department of Biology at MIT, and he co-founded the company with Professor Chris Voigt, also from the Department of Biology.
All right. Thanks, everybody, for being here. My name's Alec. I'm a co-founder of Asimov, which is a new synthetic biology company spinning out of Chris Voigt's lab at MIT. And I'm founding this company with my classmate, Raja Srinivas, who's also out of MIT Biological Engineering, and my thesis advisor, Chris Voigt. And so we're a genetic circuit design company. And so the big picture idea is that everywhere you look in nature, cells are performing exquisite feats of biochemical computation. So here I'm showing you an illustration of the membrane of a macrophage, a human immune cell. And you can see it appears sort of like this chaotic molecular environment. But despite the chaotic appearance, it's actually performing very sophisticated biochemical computation using DNA-encoded biomolecular networks called genetic circuits. And so literally everywhere we look in nature, cells are using genetic circuits to communicate with one another, to self-organize into spatial patterns, to navigate their environments. And this is in contrast to the state of the art in biotechnology, where we simply overexpress one or a handful of genes. So imagine what we could do if we could engineer genetic circuits the way nature does. It would enable an entire array of biotechnology applications across materials, agriculture, and healthcare. And so people have been working on this problem of engineering genetic circuits for about 2 decades. And it's been a foremost technical challenge in synthetic biology. It's only recently that we had somewhat of a breakthrough where we integrated ideas from genetic engineering, cell biophysics, and computer-aided design. So we called it a programming language for living cells. We published it in Science last year. And how it works is a biological engineer can code up a specification for gene expression. So think the timing, the magnitude, and the conditions of gene expression within a living cell. What the software then does is it performs a series of circuit synthesis steps to arrive at a DNA sequence. So at a high level, it allows you to go from a high-level specification all the way to a DNA sequence completely automatically. Okay, and we've used this to actually build the most complex genetic circuits to date, everything from decision-making to metabolic control to memory that's readable and writable in a living cell, and some really complex cell patterning circuits. And so now we're turning our eye to industrial applications. If you plot the complexity of these engineered genetic circuits over the decades, we think it's recently we've hit a phase transition for building incredibly complex things that is only possible with computer-aided design. So the platform has been validated in diverse cell types. And internally at the company, we're developing applications in manufacturing to augment the production of biologic therapeutics, but also some genetic circuit applications in living therapies. So bacteria that can live in and on the human body, and of course, human cell therapies, which are an incredibly exciting area which genetic circuits will be transformative for more precisely sensing and responding to tumor and immune signals. So the two sorts of partnerships we're looking for right now are external, where a company comes to us with a specification in mind. So again, timing, magnitude, and conditions of gene expression for a new application, and we build a circuit to that spec. Or alternatively, we've developed a lot of circuits in-house that are potentially applicable to therapeutics. So we'd love to get these in the hands of partners to start testing and doing pilot projects. So reach out to me. My name is Alec. My email is alec@asimov, or find me afterwards. Thanks for listening.